Hello, everyone. Good morning or good afternoon. Thank you, everyone, for coming in person and online to our special seminar. It's really a distinct pleasure to be introducing Renzo Huber to you guys. Renzo is really, up and coming and very promising MR physicist who's worked at the functional MRI facility at the NIH with Peter Bandettini now for probably more than 5 years, 6 years in aggregate. Yeah. Renzo did his PhD at the Max Planck Institute in Leipzig, working with Bob Turner and others, and then did a postdoc with Peter at FMRI facility at NIH from 2015 to 2019. Then went back to Europe and was an assistant professor at Maastricht University for 2-3 years. Yes, where he was in a tenure track position, and then kind of switched gears a bit and came back to the NIH to serve again as a physicist there. And so Renzo's been a pioneer in the development of Layer-fMRI methods, so looking at fMRI within cortical layers, and I think as such has been able to find ways to marry both methodology development and interesting neuroscience questions. And I mean, I was chatting with him today and I told him, you know, your science actually, I think, was in many ways an inspiration for much of the technology development that we have all kind of seen a renaissance for at ultra high field, as well as at 3 Tesla with ultra high performance gradients. So it's a really great pleasure to introduce Renzo today and have him talk to us about his work on Layer-fMRI methods. The title of your talk is Achilles and the Tortoise, and I'm sure you will elaborate on this.
Okay. Thank you very much. It's a pleasure to be here. Thanks for having me. I also want to start by thanking my colleagues and supporters at NIH, and I want to thank Jonathan for being a source of inspiration for my entire career. And there's almost nothing I show you today that is kind of influenced by Jonathan. So you all know that Jonathan is leaving, so I thought I take this opportunity to dedicate this talk to a celebration of Jonathan's influence on my career. And I would like to do that. I would like to celebrate this with this little metaphor of Achilles and the tortoise. And maybe you know the mathematical paradox, that there is this Zen being, very wise, the tortoise, and it's a head, right? And then there's Achilles who tries to catch up, right? And wants to reach the position of the tortoise, and he does so. By that time, the tortoise has moved on, right? So Achilles tries to reach the new position. By the time, the tortoise has moved on again, and so on and so on. And he can, it's impossible to reach the tortoise, like ever. And I feel with Achilles, because I can spend my entire life following Jonathan, but at the end of the day, it's impossible to do so. There are so many dimensions in layer-dependent fMRI that you can never reach Jonathan. So Layer-fMRI is my home, the field I care about, so I want to introduce it real quickly. It's an old field. It has been around since like quarter decade. The tortoise started walking in 2010, and since then the field is basically exploding. And it's not only exploding because of better methods, but also these there are more and more application studies. So Layer-fMRI has shown to be useful, and so the red studies are the application studies. I started to enter the field in 2012 as a master's student in Bob Turner's lab in Leipzig. And I saw these kind of data from Jonathan, and I was kind of fascinated that you need physicists, I'm a physicist, in order to kind of understand non-physicsy things like the brain and the mind and so on. So I was kind of naive and thought I can contribute physics skills to understand something like the brain with all these different specializations, including kind of things that make us human. And it was an exciting time to kind of contribute to develop tools to see the brain in action like that. However, it's also somewhat disappointing, I found, to try to understand how the brain works by just looking at these kind of blobs, right? They're just blobs. And trying to understand how the brain works by these blobs is somewhat like trying to understand how a truck engine works by looking at it with an infrared camera, right? You kind of see activity, you see the big structures heating up, what consumes energy. But do you really learn a lot about the mechanics, right? Or do you know if the truck does not heat up? and you now learned that the front is involved in driving, if it doesn't heat up, how do we fix it, right? We need a different spatial scale to really understand kind of more of the underlying mechanics of that. So in this metaphor, in fMRI, we need more than just the voxels of gray matter, white matter, CSF. We need of kind of the spatial scale of the layers, right? Because these different layers, they contain different neural populations that do different signal processing. So increasing the resolutions of your voxels from something like this to layer resolution allows you to kind of subsample populations of layer groups, which gives you a lot of additional information. It not only tells you if a given brain area is involved in a certain task, but importantly, it gives you some context about this activity, like where this activity is coming from, how it's following the internal processing step, how it's modulated from feedback from other areas until you have a computational neural output that's sent to further areas with respect to feedback and so on and so forth. So with Layer-fMRI, we not only know which areas are connected to each other and which of those connections are stronger than others, but importantly, we learn something about the directionality, the kind of the causality of these networks. Now, to get to these layers, however, and see them, we do rely on decades of methodological advancements. And I would like advancements with a metaphor from Bruce's Brain Observatory talk, namely looking at astronomy. Let's look at this beautiful night sky, and we have some areas that are just brighter than others, right? And then you can zoom in and you see, oh, there are these clouds, right? And you zoom in further and you see subclouds, and you zoom in further and further until you ultimately hit the resolution limits of earthbound telescopes like LaCierre in Chile. And then with decades of methodological improvement, you see that it's actually, there's more information than just there's a cloud, right? You see these little wiggles at these contours, maybe you can see them. And with another a few decades of improvements, you can see these contours, these wiggles are not only at the contours, right? In fact, the entire cloud has these wiggles, right? And you suddenly get an additional information about the dynamics of those clouds. And I'm showing you this metaphor because this is just about the time scale of fMRI method developments, where the big leap happened in the early 90s here with very coarse resolution. And here in this example from Peter, they were like purposefully very coarse resolutions. And there is this Moore's law of fMRI resolutions that about every decade, you get about 1 order of magnitude in voxel volume reduction. And each different spatial scale has this unique window to have additional information about the brain. Like in the last 30 plus years, we learned a lot about the brain as a field by looking at these big structures, the different brain areas, right? It has revolutionized the world. However, it's still kind of an oversimplification to think that all the neurons across the entire brain area are doing the same thing, right? So with high resolution, you can even look at kind of topographical representation differences following the cortical ribbon. But still, you kind of need to assume that all the neurons across the entire cortical depth are doing the same thing. Now, with these higher sub-millimeter resolutions, you can see that you have different kind of approaches of looking at these subpopulations of neurons across the different cortical layers. And some people think of kind of 2-layer models, input, output, or 3-layer models, feed forward, feedback, or up to 9 layers. There are so many definitions of layers, and I'm using that term rather agnostically. So at these kind of spatial scales, we have a lot additional information to be seen, which I would like to exemplify by zooming into these tapping experiments here, where you see we have these kind of stripy patterns following the cortical ribbon that I would like to interpret as input-output circuits. But also we have these kind of columnar-like things that I'm not really sure what they mean, which is like the exciting part of working in this field, right? You see something new at different resolutions, and then the fun starts, and you can try to figure out if this more refers to vessels or neural structures or so on. Now, to get to these kind of stripy images, it's not enough to just increase the resolution of your readout, but we also need to worry about the right contrast mechanism. Because at the end of the day, fMRI is an indirect measure of neural activity, which is described in this little cartoon here, where we have the neurovascular coupling. You see that neurons fire and they trigger this little arterial vessel kind of to open up very locally. We have a blood volume increase, and a short while later also we have these oxygenation changes in the veins coming through. And you already see the problem, right? That these oxygenation changes in that vein are not only representing the neural activity close by, but this vein is coming from somewhere outside that voxel, right? So we have a localization specificity problem, which is well known since the advent of fMRI, that when you just take a conventional gradient board and increase the resolution further and further and further, you have these false positives at the superficial layers, which means that you end up with something that still looks like a kind of fuzzy blob. Now you have this fuzzy blob at very high resolution, but it kind of, due to the surface activity, is still kind of a fuzzy blob. So just using gradient echo bold at the superficial layers with all the sensitivity as a measure of layer activity is somewhat like using CO2 emission mapping as a measure of industrial activity, right? It works very well for the big structures, the countries and cities and continents, but it breaks down as soon as you approach the spatial scale of the training infrastructure. So you cannot use the overall CO2 emission of a voxel like this as an indication of the activity in this little building over there, right? Because many cars emit CO2 while going to other buildings outside that voxel. And there are many potential ways around this training bias. Some of them refer to different acquisition methods. And my personal favorite method is this VASO method that is believed to be mostly sensitive to blood volume changes that are very closely localized to the laminar neurostructures. And with that, I'm kind of trying to catch up with Jonathan because he has alternative methods. For example, trying to get rid of these vein artifacts by kind of going away from those veins spatially and seeing very beautiful M's without those veins artifacts. I'm instead trying to hope that VASO really measures what we think we do, but what it does with respect to blood volume imaging. And to validate the origin of the VASO signal, I had this grant with an iron cannulate to re-implement the sequence in a Bruker setup, and an iron built an endoscope that can be used inside the MRI scanner, so you can simultaneously do optical imaging spectroscopy as well as doing the VASO-MRI, and then you can basically place your slice right on top of one of those veins that you directly see. And with gradient echo bold, you have this kind of vein at the surface over there. With VASO, you don't. And because it's animals, you can also inject the iron contrastation-based fMRI methods, which gives you a different flavor of blood volume in different units. If you normalize it to the right units, it does look like VASO can be specific to these blood volume changes that are, in some cases, localized to the laminar neural structures. And in trying to understand the VASO contrast, I'm trying to catch up with Jonathan, who is also now looking at understanding the VASO contrast with Grant's work of simulating the rodent physiology.
Just for one.
Yes.
So looking at the bottom three images in the middle,
Right.
No, they look more similar perhaps than they look to the gold, but they don't look super similar. The figure you show above, they really match remarkably well.
Right, and which is why I'm showing the percent signal change down there. So that's why I said it's a different flavor of blood volume. So Mayan is kind of really changing the baseline signal, right? In Mayan, you have when they're macro vessels, even though they are not kind of dilating the large veins or even the arterioles, they kind of lower the signal at the superficial layers. Then Mayan is inherently sensitive to the relative blood volume change. And because we have high variance or heterogeneity of the baseline blood volume change, there's much more baseline blood volume in the superficial layers. It kind of weights down. Like if you look at delta CBV over CBV0, the denominator is huge in Mayan. In VASO, we are not measuring relative blood volume change. We are measuring blood volume change in units of percent of the tissue voxel. So it's kind of the same denominator across all the different layers. So basically here we are kind of missing the superficial layers because the baseline blood volume is so huge in these superficial layers. And that's why we kind of need to look at it in milliliters per tissue volume and not we need to look at it and normalize it to the same reference. If this is kind of the baseline blood volume, which is heterogeneous across steps, or if it's kind of the voxel volume that defines that. Which took us a long time to figure out these discrepancies, yeah.
It's so simple.
Yeah. And we had some monkey experiments later where we could confirm that in Lokotitis lab with Josine Gönze. So what is the kind of additional information we can obtain at these spatial resolutions that we cannot get with conventional imaging at conventional resolutions? I would like to show this little example here, again, looking at the motor cortex, where I'm just doing a finger tapping experiment, and you can see the sensory motor system kind of lights up, which is cool. Now, if we try to make this more neuroscience-y, we can also ask the person to just think about moving their hand, which has weaker activity. It's skewed a bit differently, but overall, it's kind of frustrating how similar the brain looks given this very, very different behavior. So then we can zoom into the mesoscale regime and then decrease the resolution further and further and further until we see these stripy patterns, where we then have these two stripes in this very thick motor cortex, where we would like to interpret these superficial layers as kind of cortical integration feedback layers, where we have motor planning input, sensory feedback, and so on. Whereas the deeper layers have a lot of like these output recurrent circuits that ultimately trigger the kind of have these directions to the spinal cord and trigger the muscles to move. Now at these spatial scales, if we look at the imaginary tapping, we don't really have this secondary output stripe like at all. We have still the computation going on, the same areas involved, but it's involved in a different way. And this is kind of the additional information that layers can give us. We were trying to look at focal hand dystonia patients. Those are people that behave normally in everyday life, but for some tasks, they cramp, like for writer's cramp or a musician's cramp. We looked at the writer's cramp focal hand dystonia patients, which are kind of mesoscale naive participants. So it was a bit challenging to get kind of robust results and then kind of teach them to lie still, so we could get these consistent tapping experiments like with the high resolution and the long scan times that we need for that. And what we did find was that for the affected hemispheres, we have again this kind of imbalance between input and output, where for the unaffected hemispheres, we have this nice reproducible like double stripe, a double peak pattern, whereas for the affected hemispheres, kind of the deeper ones that are lower, whereas the superficial ones are shooting up. And Mark Hallett from NIH was very, very happy about this result because he had this theory of that the focal hand dystonia might be caused by a lack of surround inhibition, which we would expect in the superficial layers. And therefore, kind of the neighboring fingers are kind of all activating each other, and then you end up cramping. So with the mesoscale tools here, we can try to start to test these hypotheses that were not testable beforehand. The kind of lessons that I learned from these experiments and application studies were that it's not really trivial to do layer-dependent fMRI without doing columnar imaging, like without looking at the topography of the activity, because these double stripes are there if you know where to look for it, for these pinch movement part in the lateral side of the primary motor cortex. So in order to develop this with the messy data of layer-dependent fMRI, where we usually only have kind of thin slabs, they are distorted, and they don't fulfill the common topology requirements that you would need to have for mesh-based layerification of closed surfaces without holes, we tried to develop this again in the test bed of the motor cortex, looking at digit representations, which I would like to show you here, and hopefully this works. So it does not. I apologize. I will try to fix this. I just need to find my slide. There's my slide. And now, okay. I have two screens now, so my shortcuts don't work. I can do it differently. So basically, we have the central sulcus over here. The motor cortex is much, much thicker than the sensory cortex. You see that? And then I'm basically using a series of flooding algorithms to like select voxels as kind of voxel borders, basically, as surfaces, first in black for the sensory cortex, and then in light gray for the motor cortex. And then basically you can flatten those patches of the cortex in the tiny slabs that we have in EPI space and then look at it in 3D. And basically now I'm trying to rotate this to look at it from the perspective of the back of the brain, where the midline would be somewhere here, the right ear would be somewhere there, and then I'm projecting the individual fingers onto it. This would be the pinky finger, the ring finger, the middle finger, index finger, and thumb. And in the sensory cortex, that's kind of well behaved. It's kind of almost macro scale. Neighboring fingers are next to each other in the brain, and it's well behaved. And it's huge, like a huge chunk of the central sulcus is occupied with kind of the individual finger representations. Now, more interesting is what happens on the other side in the motor cortex, which is kind of harder to measure, specifically also in conventional gray neck bolt, where there's venous signal leaks. And I'm looking here in the hand knob, and you can see again the pinky finger, 2 ring fingers, 2 index fingers, and 2 middle fingers and 2 thumbs at the end. So in the motor cortex, I had to scratch my head. It was kind of messed up, right? It was, first of all, it was smaller, and we had multiple mirrored representations, almost like reminding me of the mirrored representation of eccentricity maps between V1 and V2. And these representations were tiny, like the entire hand representation here is as thin as 4 millimeters or so, which might be better appreciable if you look at a kind of projection like that, where you can see that at the bottom left in the sensory cortex, we have this huge somatotopic behaved finger representation. On the motor cortex, we have these weird mirrored representations that are tiny. And I was surprised because I didn't see that in the fMRI literature. However, when you go back to Penfield, or the homunculus model was mostly based on the sensory cortex. In the motor cortex, he already had shown multiple representations in there. So we tried to tease apart these different representations, for example, with different movement actions. Like, for example, when you do grasping versus retraction movements, we can see that we end up with patches that really nicely match the borders of these multiple finger representations. So in the motor cortex might be a body part representation map for the individual spines, but when you look at the mesoscale, it doesn't really make sense to think of the cortex as representing fingers. It represents movement actions. So it matters if you use the same finger in one direction with that muscle or the other direction with that muscle. And this was also highly consistent across multiple participants and has been followed up by other people, also looking at kind of different body part representations. But you cannot only do layer-dependent fMRI vaso in the motor cortex. In fact, Jonathan started in the visual cortex, so I need to catch up, right? And visual cortex is hard. It's thinner. It's highly convoluted. Yeah. Yes, they were. Like the extension reflection are kind of the different colors here. And it was still weird. Like, for example, sometimes we had extension reflection kind of medial to lateral, and in some of the cases was the other way around. And it kind of also depends on how deep you are in the central sulcus. So we were kind of really at the kind of top part, the evolutionary older part of M1, where, yeah. But the borders always matched with the retraction versus extension, like this little toy model. And those are differential maps, I should say. So it's not that one patch only reacts to grasping and the other only for retract. Those are like, they always react to everything, but one is stronger with one compared to the other. All right, visual cortex. At NIH in our lab, we have Sharif. Sharif is a consciousness researcher, and he cares about his paradigm of after images, where he shows kind of faces, and then people don't really know if they have an after image from their eye or if they're actually seeing something or not, because it has been so oversaturated from the inducer, the black face over here. So we are very interested to measure at these different conditions with Layer-fMRI vaso in the primary visual cortex, where we do see that for the inducer in turquoise, we have like a lot of activity in the middle layers, even more so in superficial layers. And this kind of shifts as we turn it around and look at the after images, as opposed to the mock after images, where the deeper layers are actually the ones that shoot up, and the feed-forward middle layers actually are reduced. So there seems to be different kind of mechanism involved that triggers these after images from kind of higher cortical areas down there. But Jonathan not only looked at kind of layers, but also was helping Shaheen to look at columns. So I'm trying to follow this with helping Alessandro Pizzuti to look at columns in the visual system, specifically in human NT plus V5, which is known to have these axes of motion columns, where you basically have dots moving in different directions and the different tuning curves for different sequences. And in this small patch of the cortex of V5, we see that we have these different representations of the columns, which were nicely behaved in the sense that when you take the small patch and then flatten it within the slab acquisition that we have, you can see that most of the activity kind of spans across the entire cortical depth, and it's nice interdigitated. So it's not kind of big blobs that are dominated from vessels, but they are kind of right next to each other, interdigitated. Staying in V5, we can also try to look more into kind of pathology, if that's the right term, like looking at blind people, where with Marco Barilari from Liège, scanning in Liège, he was at NIH looking at those data until a few weeks ago, looking at sighted people versus blind people with very exciting preliminary results, where you see that for sighted people who are not blind, when they have kind of visual motion paradigms, V5 is activated, as it should be. When they listen to auditory motion, they see that V5 is actually suppressed. Now, when we look at the blind people's data, we do see that it's actually the other way around, as is known from lower resolution, that V5 is suddenly becoming activated for kind of auditory motion perception. But interestingly also, it really seems to be a feed-forward driven, like looking profile, where it looks like due to plasticity from these people who are blind from birth, that they are basically processing auditory motion by using their processing streams and kind of taking over some of the ones that we would consider visual, which might be just like motion sensitive areas like V5 and MT. But visual and motor systems are not the only areas where you can do layer-dependent fMRI. You can also try to go more and more cognitive. For example, here in the dorsolateral prefrontal cortex with Emily Finn, looking into this tiny patch of the cortex where we do see that for different task conditions, we can differentially activate some layers more than the others. For example, having these strings of letters and then asking people to kind of sort these letters according to the alphabet and memorize that order really seems to engage these superficial circuits over here, the delay cells, as they call it in the monkey literature, as opposed to the response cells, when you later need to do a response decision based on the location of that letter, which then mostly engages the deeper layer, sending the output to the motor cortex. One issue with all of those different applications is that we are really zooming into these tiny patches of the cortex and then looking at the layer-dependent modulations, which is kind of not really fulfilling the full potential of layer-dependent fMRI, because in layer-dependent fMRI, ultimately, it's a connectivity method, right? We are looking at input, output, feedback input, feed-forward input. But looking at these tiny areas only doesn't really tell us where this output is going to, where this input is coming from. So we need to go away from these tiny little areas, but follow Jonathan and go to networks, which was a hard problem for me. And I invested at least the last 5 years on it to not only expand it to entire slices and slabs, but then ultimately also go to like enough slices that you can, which you can call kind of whole brain layer fMRI at the cost of TR. And in the first iteration of trying to get there, I was mostly interested to just see if there's signal that we can harvest. So here looking at the common kind of networks for a movie watching paradigm, where you can see that there is signal. In fact, like it's for the default mode network, we have this kind of one layer following the cortical ribbon. And in some networks, like here, these frontoparietal networks, we have these double stripe indications as in the interparietal network, which means that there's a lot of potential for cognitive neuroscience connectivity analysis, who usually, those folks usually chunk up the brain in all these different brain areas and then look at the correlation of each area to each other area and look at these connectivity matrices. Now, with the additional information of layers, you not only know if they are connected, but importantly, you can infer kind of the directionality, the hierarchy of these connections. And to validate this, I followed Jonathan, looking at 2010 abstract of looking at V5 and V1. And this kind of assumes that in hierarchical brain system, any given brain area receives feed-forward input, preferably from these lower hierarchical areas into the middle layers, while receiving feedback input in the same area, mostly in upper and or deeper layers, from other areas that are higher in the hierarchy. And then having localizers, functionally and structural localizers of kind of thalamus, V1, V5, we can try to do kind of seed-based analyses and look at the layers of V1 while using different seed regions. For example, having seeds in LGN and a bit of Parvina, we do see that mostly the middle deeper layers of V1 are synchronized with those fluctuations. The superficial layers in V1, they don't really care what LGN is fluctuating about. This is different when we place a seed region in V5MT, where we have this additional feedback at peak here close to the surface, which might refer to these off-diagonal elements from Jonathan there. So now that we know that layer fMRI connectivity gives us signal in the whole brain, yes.
So is it resting state or is it no response to movies?
This was resting state. Yes.
That's what it's
This was resting, yes. Yeah. And this was actually a slab only. Like this was just to test the signal. This was not the whole brain.
What
Yeah.
was the duration of your scan?
It was 3 times 15 minutes.
3 times. Oh,
Yeah.
okay.
This is even longer. This was 13 sessions of 50 times watching the same movie. The purpose of this study was to basically optimize whole brain layer fMRI with respect to kind of the best undersampling pattern as well as multi-shot methods, but importantly also to kind of characterize the stability. So this is the same movie watched over and over again, and we do see that we can get kind of consistent results. And it was mostly kind of acquisition-based study. And still, even though it was kind of a sequence-focused study, the data that are on OpenNeuro, of course, have been downloaded like crazy. So I think there's this huge need in the cognitive neuroscience community to look at whole brain layer fMRI connectivity. So I think, and now that we have a method, we know that there's signal and it's stable, we are ready to start the whole brain layer fMRI 3.0, which I think is something I want to do in the next 5 to 10 years, to do these deep sampling, measure the kind of same participants, like a lot, to not be limited by SNR, and then actually have something to give cognitive neuroscientists, not just kind of something that was optimized for methods. Yes.
To what extent it was consistent if you were doing this?
Like with this deep sampling, I didn't do resting state because it was long enough already. I wanted something that I can average, and I wanted something where I can also look at the differences, like how things change across the different movies, for example, in entorhinal cortex with memory and the time scales.
I understand
Right.
that, but the problem here is that
Right.
majority of our studies are based
Right.
on resting. So that this one, signal is mostly driven by stimulus. So
Right.
you're saying that it's stable because of the consistent stimulus that you're presenting.
But I didn't really care about how stable the brain is. I cared more about how stable my acquisition is, which is what I mean. So that, therefore, I think that data set, even though it's downloaded a lot, it's not the right data set for the questions that neuroscientists are using it. I wanted to see the stability of my sequence, given all the artifacts that I had with these low bandwidth and multi-shot approaches and all those kind of things. Yeah. There's a follow-up study from Yuhui Chai in Illinois, who has been not looking at movies, but almost the same coverage, looking at resting state only. And he does see quite consistent results with a slightly different sequence. All right. In fMRI, as in layer-dependent fMRI, we are always kind of constrained to this triangular parameter space. We can have coverage, high resolution, or fast sampling. But as soon as we push one of them, we kind of need to make sacrifices with the others. And I already told you how we can kind of try to improve coverage. Let's see how much we can push it with the other angles. For example, looking at resolution, following Jonathan and Avery's multi-shot approaches, which in 3D EPI is actually not too complicated and rather straightforward. Especially, I'm lucky to and privileged to use the 3D EPI sequence and have full control of all the looping structures, including the vaso and then the corresponding reconstruction, where you can get to these 0.1 microliter resolutions with kind of any protocol that you want, with vaso, without vaso, doing it single echo or multi-echo, with quite exquisite EPIs, as you can see them in the underlay. And this is on the Terra, where we're really encoding limited based on the gradient performance. If you combine this even with kind of more advanced gradients that are being co-developed here, I was scanning on the Fine-Bergertron scanner for a long period, always Monday, remotely, to even push these resolutions further and further. And in the visual cortex, which is very thin, then to also kind of have Nyquist samplings across these different layer groups, for example, to have indications of these multiple stripes, as well as these kind of bimodal patterns that I'm not really sure what they mean yet. If you don't care so much about these very high resolutions, but instead want to be fast, you can try to follow Jonathan, what he's doing with Daniel Gomez and Laura. I'm trying to do this with Sebastian Dresbach, a PhD student, to use a sequence where we have very short kind of volume acquisitions in a small coverage, looking at checkerboards of variable duration. But on top of that, also having these very high resolution multi-echo GRE data at 0.35 millimeter resolutions, where you can directly segment the vessels, like the peel vessels, as well as the diving vessels. And at these kind of temporal sampling approaches down to kind of 1 second stimuli, we do see that, like for conventional block designs in gradient echo bold, if you look at the big vessels in red here, we have much stronger response compared to the micro vessels in layers. For vaso, it's kind of the other way around. We still have signal of the macro vessels in red, but most of the vaso signal is dominated from the micro vessels in the layers. As we go to shorter and shorter stimuli, we do see that this is kind of changing, that the micro vessels, they react very promptly in blue here. They always have the fast onset, but then as soon as they dilate, they don't dilate so fast. They take their time, they're sluggish, they're passively controlled downstream, and kind of capillary, presumably, vessels that are not kind of actively controlled with smooth muscles. And it takes time for them to accumulate. You need to give them like 20 seconds to really outperform and overcompensate the macrovascular components. So at these fast samplings, we see that the trunks of the arterial vessels, which do dilate, as we see from optical imaging, they start to dilate later, but as soon as they dilate, they really shoot up. And then they are pretty quickly kind of saturated. And so kind of as you go faster and faster, you have a different window of looking at these kind of different vascular compartments, and you end up with actually more macrovascular contamination also in vaso. It's not half as bad as gray nickel bolt, but still it can be quite annoying. And this is something where I really appreciated these kind of high-resolution structural reference scans. And I think high-resolution vessel segmentation is really something that the field needs as a standard tool in the next 5 to 10 years. All right, while we are constrained in this kind of triangular parameter space, I think sometimes we can cheat a bit and push all of them at the same time and just get away with more SNR by following Jonathan. And namely, he introduced these kind of surface smoothing to the Layer-fMRI domain, and which is something that I think I saw in 2015 at ISMRM, and later he followed up with Anna on this. So as soon as I came back from that ISMRM, I also implemented this in my little Layer-fMRI software suite, Laney, in voxel space, where you can kind of sacrifice completely the columnar dimension, but then you get very explicit results, even in very, very noisy data by kind of smoothing only within given layers without signal leakage across different layers, which can be valuable for some applications. And this Laney is a software suite that I started and is now mostly maintained by Farouk Gulban, and professionalized and added. And recently he wrote a book chapter about it and found that for the single purpose of within the slab doing layerification, this has now become the most popular software suite out there. And with layerification, I mean this very hard problem of kind of predicting a layer location based on a kind of a segmentation input. Indicated here, where we basically have as input the gray matter, white matter, and gray matter CSF borders, and you see the stria genari over here, and you see how it's kind of closer to CSF at the crown of a gyrus as opposed to closer to white matter at the bottom of a sulcus. And in conventional Layer-fMRI, I think it really doesn't matter. But if you just take the difference of the distance, you kind of get a good estimate, but it's not perfect. And there has been some lobbying for people to use this so-called equivolume approach, which can kind of predict this specific layer with a slight more accuracy. And I think that's the reason why some of the people use it. And let's talk about the future. What are the things that Layer-fMRI that still hold us back? I think it's mostly like, I think Layer-fMRI is just starting and becoming useful, but ultimately to fulfill its whole potential, we need more than the cortex. Like there are all these subcortical cortical connections, and we need to have a complete picture for Layer-fMRI really to kick off. And it's still limiting to do anything kind of in the bottom half of the brain. So if I divide the brain into two halves, only 8 out of 250 papers that I keep track of on my blog are looking at the bottom part of the brain. And this is because it's hard, right? There's low TSNR, transmit efficiency is just challenging. Distortions are bad, but I think right now the most limiting factor is the EPI, like matrix size. You cannot use these tangential field of views. You need to have huge matrix sizes of 300 or so to have decent resolution, which is challenging, right? A conventional EPI-based fMRI, and you acquire your K-space going back and forth and back and forth and back and forth, and you just cannot do this 300 times and still have signal. So conventionally, people kind of skip K-space lines and then try to fill in the missing lines by other means. And a prominent approaches would be grappa, but it has its limitation ultimately, right? You cannot do this to like really, really, really huge matrix sizes. So alternatively, or additionally, you can also kind of combine different TRs with different KY prefaces and combine those as a sliding window or a segmentation approach. So you can get the same images with the same trajectory and the same blurring and the same distortions, either by using no grappa and a lot of segmentation, or a lot of grappa and no segmentation, or any combination in between. And if you do a lot of segmentation, we are in the physiological noise dominated regime because all these shot-to-shot variations, and therefore you are not really TSNR efficient because of the physiological noise, which I think is a powerful message that even at these resolutions, we are not at the physical limit of the thermal noise dominated regime. As you go to like overdo grappa, we are kind of encoding limited from the coils and have artifacts. And if you go a bit more easy on grappa, you still have the G factor, and the G factor is something you never get back, right? Even if you have more TRs, there's always noise amplification. So there might be this optimum dependent on the protocol and area of combination of segmentation and G factors, which is, I think, a subfield in layer-dependent fMRI that I want to spend more time on. And I think most of the innovations in high resolution acquisition in the last 5 years are coming down to an optimized combination of subsampling as well as multi-shot methods, and many of them from this building. The second big issue for high-resolution EPI, specifically in the deeper structures, are the things that I call the fuzzy ripples. So not everybody who publishes Layer-fMRI makes their data publicly available, but those who do, I downloaded all of them, and in every single study, I see these kind of low spatial frequency fuzzy ripples. Specifically in the deeper brain structures, where you not only see these off-resonance effects, but also these kind of low frequency shading patterns. As you go from 2D to 3D, these off-resonance effects kind of average out or blur away, on top of the benefit of the higher SNR, but these kind of low spatial frequency fuzzy ripples are still there. So one approach to account for this that we have taken is to basically use a dual polarity EPI to basically isolate those artifacts and have them with a complete opposite phase, and then combine two different TRs with the opposite phase as a kind of synthetic interleaved flyback EPI, which is an approach that I found very convenient because you can do this directly in projection space in the Siemens ice reconstruction. So you don't need to tell users to download raw data and get rid of these artifacts as well as the off-resonance effects. And this is following somewhat what Jonathan has been doing with respect to the dual polarity grappa approach, which takes care of these off-resonance effects and even kind of nonlinear phase errors a lot. It might still have some of the low spatial frequency trajectory error related artifacts in there. The kind of most efficient approach to deal with those kind of artifacts would be to avoid them in the first place, right? And for that, we need to isolate where they're mostly coming from. So let's look at an EPI gradient trajectory, the nominal one. And if you measure it, you can see that we have these eddy currents and we have imperfections. And these imperfections are kind of have a specific feature. They are really at the borders of the ramp sampling. And in EPI, high resolution EPI, we do a lot of ramp sampling, usually because we're encoding limited. So we have these great errors in the trajectories that are very sparse in K-space. So they cannot be globally applied with one single grappa kernel. It would need to be a specific KX specific grappa kernel. So we end up with gradient trajectories and K-spaces that don't look like that, but instead they look something like this, where we start slowly and without any errors. As we achieve the flat top, we have a delay. We never really catch up, have another delay, and go back and forth and back. And so we have this ghosting in the low spatial frequency patterns. How can we mitigate this and reduce that? So these ghosting patterns then result in these fuzzy ripples. And the difference between the two images that I'm flipping back and forth here is a very simple cable at the back of the scanner that you just disconnect. It's the third order shim, which has based on, because it's a third order, it has some symmetries with the gradients itself. It's between the primary and secondary layers of the gradient. And unplugging this just makes them completely go away. And it's frequency dependent how bad those artifacts are in the first place. But it's a very simple hack on how we can kind of go easier and easier without kind of making the life harder for the user applications. Getting a better feel for good results, going deeper and deeper into the brain. I've been advising Lonique Fais here to do auditory layer-dependent fMRI, which inherently might be useful with VASO because you have these silent gaps with the inversion delay to get kind of consistent, reproducible layer effects in there. And in advising Lonique to do these kind of auditory layer-dependent fMRI, I'm just following Jonathan in how he helped Aveninen to do a layer-dependent fMRI in the motor cortex. And getting kind of more control of the readout allows you to kind of do a lot of kind of readout gymnastics that might be fun. So here, for example, more currently with Kenny Chung, a technologist at NIH, I'm trying to use a 3D EPI as a faster version of MP2 Rage, which is kind of something that almost all of our users use. But using EPI has two issues, right? Specifically the distortions, but also these kind of phase inconsistencies, trajectory errors when you do too much ramp sampling. So we try to account for this with T1, 2, 3, 4, how we call it. It's T1 weighted scan with 2 inversions, 3 EPI, and 4 readout directions to really kind of separate those artifacts separately. And then also because it's kind of well behaved in orthogonal directions, you can also do this directly in the ICE recon and end up with kind of these artifact 3 MP2 Rage like images that you can directly push through FreeSurfer with any kind of distortions that you want to have in case you want to match it with the structural acquisition. And because like this entire thing takes about 3 minutes, but it's a few averages, the individual volume TR is 20 seconds, which is just approaching the time scale of fMRI, right? So you can acquire fMRI scans right during your structural acquisitions. You can use the second inversion time for bold imaging and then use the complex valued division for uni images, which you then pump through FreeSurfer. And so we get a visual activation here. And the kind of surprising thing that some people here, including Natalia Zaretskaya, has looked at is that we have this change in T1, activity dependent. And then it looks, I'm not sure how well this comes across. On the bottom left, you see the visual cortex, the surface given from the white matter, gray matter, right, from FreeSurfer, where you see that kind of gray matter seems to dilate, and weirdly, it seems the gray matter, white matter border that seems to shift. The gray matter CSF border, not really clearly, whereas on the bottom right, in a kind of frontal cortex control area, you see there's like, you get a feel of the kind of variance that we have, but it's not really clearly constricting or kind of dilating to that matter. So giving us kind of a tool to look at these kind of plasticity changes. Again, following Jonathan Polimeni's work with Wille on kind of functional T1 mapping. And I'm almost, this is the time. And maybe I skipped this. You can't, no, I don't want to. So I think we need to be more agnostic with respect to field strength. Like on my blog, I'm keeping track of all the 7 Teslas around the world, and they're not growing so fast. Like they double every 6 years. Layer-fMRI doubles every 4 years, right? So to meet the demand of Layer-fMRI, we need to expand beyond 7 Tesla, where by now about half of the scanners out there have my Layer-fMRI 3D EPI sequence installed. But if we would go to 3 Tesla, we would have much more users. So with Martin Krombichler in Salzburg, I was looking at motor cortex experiments and modulations of the superficial layers with IPSI and contralateral tapping. And kind of the take-home message, sorry for the speech here, you can do layer-dependent fMRI at any field strength you want. It might be more cost-efficient on one compared to the other. And 9.4 Tesla that I had available in Maastricht might even like allow you to break records, but only if you have kind of the engineers working with you and making it stable. 7 Tesla seems to be a very cool sweet spot for these population studies. If I made you excited about layer-dependent fMRI, I want to make you aware of a few communities. For example, at NIH, my little Layer-fMRI working group, and we are organizing regular international seminars every other week that you can join. Also, together with Luca Visioli and Tyler Morgan, I'm organizing these layer dinners. The last one that I organized is a specific one where we looked at the interaction of Layer-fMRI with industry. And the next one later this year will be about Layer-fMRI with translational research and those Layer-fMRI applications. If you're new to layer-dependent fMRI, there's a lot of resources on the blog over here. And if you're already into Layer-fMRI and just want to be kept up to date, you can follow this Twitter handle. And as part of the ultra-high field study group, I think Marta has left, I will be the study group and brain function that I will be chairing in the next year, and Marta will be for the ultra high field study group. We are organizing this ultra high field fMRI workshop in exactly 1 year. So mark your calendars and come to that, please. I would like to wind down with this metaphor that I think Bruce showed in 2018 when you were visiting NIH and I was in the audience, the bridge of neuroscience, which bridges all the different spatial scales of the microscopic scale of synapses and neurons, all the way to the macroscopic scale of behavior and the brain areas. And it's kind of the mesoscopic regime that's still somewhat underrepresented. And these new optical methods allow you to measure hundreds and thousands of neurons at the same time. And these high-resolution fMRI not only give you brain areas, but distributions within brain areas. But the keystone that will bring everything together and allows us to build stably on top of that is the spatial scale of the basic neural building blocks, the layers and columns. And the kind of things that we can build on top of that are like multifold. There have been a number of review articles specifically highlighting our Layer-fMRI tools, for example, and the influence or impact on neurodegenerative diseases or on predictive coding models on psychosis or ALS or just general like neuroscience. And weirdly, there are more review articles about the impact of layer-dependent fMRI than there are data. So I think we are ready to get those data. We need to get. And with this, I want to thank all the people who supported me over the years, and I thank you for your attention.